So our project is about replay-based fast and statistical sequence learning with recurrent neural networks. So basically the general question we're trying to attack in this project is how is sequence learning implemented in the brain? And more specifically, we're asking two separate questions. So the first is online gradient approximation the only approach for biological plausible sequence learning? So we know there are a lot of learning algorithms for RNNs that they are like online approximation of BPT, right? But also we're asking the second question, that is backpropagation through time a proper analogy for sequence learning in the brain? So we know BPTT is notorious for its requirement of external storage of hidden unit activities. But here we argue that BPTT could be possible in the brain based on two key observations. So the first is that the replay, either forward or reverse replay in the cortex and famously in the hippocampus, is crucial for learning, right? And the second fact is that the reverse temporal propagation of hidden activity is essential for backpropagation through time. Based on these two observations, we proposed our approach, and that is the brain may approximate backpropagation through time with two separate recurrent neural networks instead of one. So in this approach, in the first step, the rewarded sensory sequences induce fast sequence memory in RN1, and then the reverse replay of RN1 serve as two purposes. The first purpose is that it leads to fast learning of forward replay of itself, and that is one-shot learning. But also is capable to train a reversible recurrent neural network, the RN2, to perform the task, to learn the task gradually, and that is the statistical learning. So together, RN1 and 2 form a local variant of BPTT. Here we'll start with RN2 first. So RN2 is a reversible coupled recurrent neural network with competing projections. So unlike previously using models with just one group of neurons, here we have two groups of neurons. So neuron group A and B, and they are mutually connected with two pair of connections. So the intuition is simple. If the red connection is on average stronger than the blue projection, then the whole network will be driven to run in the clockwise manner. But if the blue projection is stronger than the red projection, then the whole network will run in anti-clockwise manner. And that is the intuition behind the reversible, behind the reversibility. And there are multiple choices for the activation function and the dynamic equation you can use for this neural group A and B. And here we just use the simplest one, the rate neural network model with two branches for projections. The competing or the balance between these two projections determine the running direction. And here is just, we sampled 10 neuron firing rates from a 200 neuron network. And as you can see in the reverse running phase, the firing rate is perfectly reconstructed with the firing rate in the forward running phase, but in the flipped order. And if you compute the angle between the neuron pattern vector between the forward and reverse running, the angle is always near zero as the cosine theta is near 1. So based on this architecture, the gradient propagation is plausible because we only require local information in the reverse replay phase. We can compute the history as we need. But this network cell itself is incapable to deal with inputs, because if you look at the computation graph, the unfolded temporal computation graph, you find that in the reverse replay phase, neuron group A and B here do not have knowledge about the inputs in previous time step. And because of this, we need another neural network to deal with inputs. And that comes the RN1. So RN1 is about fast reverse replay with also competing projections, but the architecture is slightly simpler. So this neural network, it receives two branches of projections originating from itself, attuned by the common gating coefficient gamma. So the gamma can be generated by some external oscillating signal or itself. These two combine the projection capital F and capital G, they build a line and point attractors in the state space. Basically, it's a variant of the continuous time Hopfield network. So this network in the end combined arbitrary input patterns as a classical Hopfield network does, but in the temporal domain. So it learns different input patterns together and then associate them together and then play them back in a reversed order. This network only requires Hebbian learning rules, and in the reverse replay phase, it can do two kind of things. So first, if we amplify the learning rates of these two weight matrices, then the whole network in the reverse replay phase will learn the reverse of the reverse replay, and that is the forward replay of the reverted sensory sequences. But also, you can just generate those reverse replay patterns and feed them into the RN2, and then the RN2 can learn the task finally. So this is the general workflow. So the animal will initially the animal will randomly explore the environment and generate some trajectories which lead to the reward. And the reward will trigger the learning in the first neural network. This RNN can either learn the fast forward replay or can be used to generate patterns used to train the reverse replay in RN2. Then the RN2, based on our second slide, can perform the BPTT-like training and finally master the task. So we test this workflow with a simple T-maze navigation task with different Q type indicating different reward prediction either in the left or right end of different branch of this T-maze. And turns out that our model can successfully learn the task. So to the end, in this project, we propose a new type of sequence learning system of recurrent neural networks, which only requires only local information to perform gradient descent, and they bridge the gap between the observation and hippocampus to bring those replays, and also the long dilemma in the biological plausibility of BPTT. And yeah, basically that's all. So this I was previously thinking that time was intense, so I basically ignore all the details. So if I have questions about that, feel free to ask.
No worries, you have time. But if you want to continue, you can continue. I can wait for 1-2 minutes to have a question.
Okay, so I have a few things I want to mention here in this slide. So for this neural network, the red projection, we can call them forward projections. So the red projection, they are just initially randomly initialized. So the whole network will randomly run in the forward running phase. But those blue projections, they will try to cancel these red projections, which makes the idea of detailed synaptic balance, right? And since this GA, so if we directly look at the equation, the task of this GA and GB, their target is to cancel this FA and FB in order to implement the anti-clockwise running. So if you consider the simplest case, the linear case, that is the forward running, they're just a linear transform. Then the backward matrix, they're also linear transform. And you can see here, there exists an analytical solution for these backward projections to cancel this forward projection. And that basically means the whole network will learn the reverse replay within very short time because just a linear regression. So that's how the reverse replay is implemented in this in RN2. Yeah, so that.
Thank you so much. Thank you so much, Huzi. I have one question about Jules that he asked: would it be possible to alter or lesion one RNN while letting the second one as it is? What would be your behavior prediction? That's an interesting question. So basically, if you do a lesion study here for this architecture that you are working.
Okay, so basically this model matches some lesion studies. So for example, you can view this RN1 as a fast cache, which can be hippocampus, right? And the RN2 can be a general model of the cortex. So if you separate hippocampus and the cortex, you'll find that the whole network, the whole system is capable to perform like the fast learning, but it's impossible to consolidate those memories, transfer them into knowledge that can last for a long time, because the waves increase and decay in a very fast way in the first RNN, and that makes it impossible to learn those statistics in the task.
And if you do virtual lesion, of your model and still stimulate it with the same input, would you see the kind of distortion predicted by the lesion study?
I haven't done that, but I'm pretty sure that if we just separate them, but the RN1 is still capable to learn a task. But if the task is changed a little bit, RN1 need to learn the task from beginning. So it has no knowledge about the statistics. It can just remember the trajectory.
So Huzi, for the training this network, you use a left and right paradigm, right? But how lengthy is your sequence that you're training?
Okay. Oh, so yeah. So this one is just for the demo purpose. So in the real training, the time step is near 100. So we have near like 50 time steps for those for this branch and 50 time steps for this branch. And it can be actually extended to arbitrarily long as does not like jump longer than the threshold that BPTT can perform, because the limit of this algorithm, because since it simulates BPTT, the
do you have a result on that?
BPTT.
That would be interesting comparison. Do you have a result on that to compare that?
Oh, you mean the comparison between BPTT and this and this algorithm, right?
Yeah.
I showed I don't have a figure to show that, but you can for this figure, I done the experiment, but I didn't show a figure here. So the learning curve is pretty similar. So they have comparable performance. The only performance loss comes from the way we simulate BPTT, and I can show here. Yeah. So in the reverse replay phase, when we propagate the gradient in the reverse order, we need a transpose of weight matrices right here, and that's the problem that's the credit assignment problem in deep learning. And here we use the feedback aligned matrix, the random, So that's basically the performance bottleneck, and this prevents us to learn really long sequences. Yeah.
That is interesting. Yes.